why I'm saying it's my favorite, again, it's a very standardized test. It has been there for 30, 40 years. There's a procedure, two procedures, national procedures, one for the AASHTO procedure and one for the STM. So I guess now you know the advantages is everyone knows how to deal with this test. It's very well known, understood, but it is advantages, again, it's time consuming. You need to do a lot of testing to be confident of your conclusion. So that means it's going to be expensive as well. So now going to the meat of the presentation is what is the procedure, how to get the endurance limit. So far what I showed, I'm not saying I'm the only one who's doing this, but what we are saying is researchers, they tackled 1 out of 2 subjects. They either went to study healing by itself, understand it very well, or try to get the endurance limit by itself. But they are not acknowledging that the healing is the main factor or the main effect of the endurance limit. So the main idea here was to combine these concepts together. How can I combine the healing concept with the endurance limit concept? So to start with, this is one of the definitions that we came up with, which is called the healing index. So basically the healing index needs 2 tests, and the healing index can be measured as the difference between 2 tests, one with rest period and one without rest period. So here, the red line and the blue line, these are 2 different tests. So the blue line is the test without rest period. It's a continuous test. I keep loading the sample till it fails. Compared to the red one is the same test conditions, except I'm applying a rest period between each cycle. And the one that I'm using here, a rest period of 5 seconds between each cycle. So SR is the stiffness ratio. Stiffness ratio is just the stiffness of any cycle over the initial stiffness. So in order to normalize it, again, all the samples will start from full stiffness, 100%, and then by time, the sample will degrade or will fail. And our definition for failure, when we reach 50% of the initial stiffness. So let's say I started with 100 megapascals. Once I reach 50 megapascals, this is going to be failure. So the 50 over 100, this is going to give me stiffness ratio equal to 0.5. So again, the definition basically is the difference between these 2 values at the failure point. What I'm trying to say here is I want to see the gain when I apply a rest period. So if I have a test like what we used to do in the last 40 years, it's going to fail after 10,000 cycles. I'm going to reach 50% of this initial stiffness. How about if I apply a rest period? How much gain I'm having? So, and this gain can be measured by this equation, which is the stiffness ratio when I have rest period, with rest period, minus the stiffness ratio when I don't have rest period. So in this case, you can see the healing index for this test is 0.9 minus 0.4 minus 0.5 is equal to 0.4. Because I'm going to depend on this definition a lot. So one major challenge that I faced is running the test with rest period for a long cycle. So for the regular test without any rest period, it can go from 20 minutes to 6 hours max, which is reasonable. But once you start applying a rest period, especially for my case, 5 seconds, it's going to take, let's say, if it takes million cycles, and each cycle is going to take 5.1 seconds, so this is going to take like 6-7 months to finish one single test. So what we decided to do, I decided to run each test for 20,000 cycles only. But trust me, 20,000 cycles is still long. 20,000 cycles means 28 hours. So for each single test, I have to run it for a day and 4 hours. And what we decided on, based on the results, I'm going to extrapolate this line all the way to the failure. Because remember the definition of the healing index, I'm interested to see what is the stiffness ratio that can be compared to the one with the continuous test. So the continuous test I run it till failure because I have no problem. I run it in a couple hours, and in this case, I ran it till 150 cycles, 150,000 cycles. For the test with risk period, I ran it till 20,000 cycles, and then I extrapolated. Some of you might ask, is this too risky? I agree. It's a little bit risky, but what I have seen from the results so far is with the test, with the risk period, after, let's say, 5 to 6,000, this line starts to level up. Because again, we are speaking of healing, there is enough healing to keep this line almost horizontal. The other challenge that I faced was I need to know what is the NF without this period. Let's go back to here. So in this example, again, I always need this line to be there to know where to extrapolate my line. So in some cases, because I was running a fractional factorial, for example, these two cells. So I have here two cells, one with 0 seconds risk period and one with 5. So it happened that in this case, I have to run only the test with 5 seconds risk period. But I don't have to run the without risk period. So that led me to a question, so where I'm going to extrapolate my line? Because again, each test I ran to 20,000 cycles only, and then I need to extrapolate it till NF, or the number of cycles they failure for the continuous test. So that forced me to build a simple regression model to predict what is the NF for each combination. So again, if you still remember this equation, so I had to build this equation to predict for me the missing cells. So I had 4 options to come up with. So the first option was, you know what, I'm going to collect all the data together and come up with one model, which will give me a general K1, K2, K3 model for all the data points together. The other option was, I'm going to keep K1, I'm going to keep K2 and K3 the same, but I'm going to keep changing K1, because K1 equation is based on the binder content and the air void. And remember, I'm testing using different binder content, different air voids. So I said, okay, I'll change K1. And the third option, I said, I'm dealing with totally different mixes. Once I change binder content or once I change air voids, that means they are totally different mixtures, and I'm going to come up with one model for each binder content or air void combinations. And the last trial that I did, and this is the one that worked out, was to come up with a different NF model based on each temperature. And let me show you here these are the 4 methods or the 4 equations that I used. So apparently the one that I selected was with the higher R square. So this is the one that I used to predict the missing cells. Again, this is not the main point, but at least to predict for me the missing cells so I can know where to extrapolate my line. So the equation, this is the recommended model. It's the NF in terms of binder content, air voids, and strain level. And then once I had this model ready for me to predict what is the NF, now I could extrapolate all my lines and measure the stiffness ratio. Remember, the Y or the dependent variable here is the stiffness ratio at NF without risk period. So after I have all the stiffness ratios, I could put all of them together, the 288 samples results, and come up with this model, which is the stiffness ratio in terms of all the 6 parameters, plus 2-factor interaction, or even there's a 3-factor interaction between the factors. So the next step now I have a model to predict for me stiffness ratio. So stiffness ratio, as I said, if you remember the definition of the healing index is 2 points. So I have to run this model twice to calculate the healing index. I run it once using stiffness risk period equal to 0 seconds and get a stiffness ratio. Run it one more time, let's say for 5 seconds, and get a stiffness ratio. Subtract both of these two together and get the healing index. So I had to do this for all the temperatures, all the binders, and come up with these lines. And basically here I have healing index versus the strain level or the deformation level that I'm applying. And I'm saying that if I have lower strain level, I'm going to have higher healing compared to if I have very high strain level, I'm going to have much smaller healing. This is the key slide in the whole presentation, is how to combine the two concepts together. So the endurance limit will happen when there is no damage incurred during the rest period. What is that supposed to mean is the endurance limit can be estimated when the healing index equal to 0.5. Why I'm saying 0.5? So let's go back to this figure. So in this example, the healing was what? 0.4 minus 0.5 minus 0.5, 0.4. And what I'm saying is the endurance limit will happen if I have the red line as a flat line, horizontal line. That means there's no damage. This sample is going to go forever. There's no damage at all. Even if there is an incremental small damage, it's going to be recovered within the rest period that I'm applying. This means that I'm going to have a full healing at equal to 0.5. So going back to here, so if I have all the lines that I developed so far, and then what I'm going to do, I'm going to draw a horizontal line at healing index equal to 0.5, and then go down and select the endurance limit for each test condition. I know everybody likes examples, so I'm going to go through one of the binders that I did and how did I use the developed procedure that I came up with. So the first part, as you remember, I had to come up with the small regression model to predict for me the NF to fill the missing cells. So these are the three equations, and this is just an example of the residual plots for one model. The R-square for these models range from 65% to 95%, so I guess it was kind of acceptable. The second stage, as you remember, after I fill all the data points, I need to come up with the big model, the stiffness ratio model. And what happens here is we use a statistical package, but basically we want to understand also what's going on during the process. So the first stage is we assume that all the factors that I used are important, are statistically significant, plus the two-factor interaction and the three-factor interaction. What do I mean by interaction is basically, is the binder type times air voids, is it important or not? Either is interaction or not. So I start with the screening stage, and this screening stage will show me based on the p-value of, or the alpha value is equal to 0.05, is it significant or not? And basically, I needed to repeat this stage several times till I come up with the last stage that all the remaining factors in the statistical analysis are statistically significant. So if you can see here, the highlighted ones are the significant factors. So I remove the other ones, repeat it till I see that all the remaining factors in my model are statistically significant. So after I develop my model, this is the model that I have. I know it's kind of scary because again, we have 6 different factors. There's also some interactions. So in order to simplify, to make it user friendly, I simplified this equation at least into 3 rows. So again, remember, this is the model is in terms of stiffness ratio, and connecting the stiffness ratio with all the other factors. So here, this is how to judge my model. Basically, this is a fundamental graph to show measured versus predicted. So this is what I measured in the lab versus what is the equation or the model is predicting, and yeah, the R-square was kind of high, so I was kind of confident with the results. So the next step, as you recall, the stiffness ratio is the step. There is another step after that, which is how to calculate the healing index. So again, I have my model. I run it twice, one with 0 seconds, one with 5 seconds, to plot the healing index, and then the endurance limit will be basically once I reach healing index equal to 0.5. I go down and I select my endurance limit based on different combinations. You can see here I have 4 different lines.